Welcome, Naama, to the Collaborative Research Center Origin and Function of Meta-Organisms here in Kiel. We are connected online. You are sitting in Haifa. It's 3 p.m. in Haifa. It's 2 p.m. here in Kiel. And it's a real great pleasure and an honor to have you here, to chat with you a little bit behind your fantastic research. You are still young, but you are already a PI. You are an assistant professor at your institution. But in these difficult times, we want particularly highlight what I call the next generation of junior of scientists, and you are certainly a fantastic example of that. We should start with the question, which is always not so very obvious. You presented and you are publishing wonderful work on the interaction of microbes with the gut of mice, and you are studying in depth the mechanisms. The question many people don't really know and they want to know is, how did you get to that problem? I mean, a key point in your career, you studied in Tel Aviv and in Weizmann, and then you went to as a postdoc to Harvard. And I could think that probably the lab of Dennis Kasper was maybe very influential in getting to your question and your niche, but I'm not sure. So how did you find this question, which is so exciting, but also which drives you now?
It's a great question, and yeah, indeed, not obvious. And I will start by saying thank you for inviting me here. It's an honor for me. So I can say that I was always really intrigued by bacteria. I remember the first lecture on bacteria during my bachelor's, talking about them as I'm quite sure it was on Helicobacter pylori, that has this machinery that with these flagella that can turn around and as a propeller, like help the bacteria integrate into surfaces and into cells. And I just thought this unicellular, only 1 micron bacteria can perform so many functions. So that was the seed, and it was really long ago during my bachelor's. Then throughout, I still was always thinking about bacteria. But somehow I turned to be in labs that, and I didn't study bacteria that much. So I was interested to explore molecular biology. So in my bachelor's project, it was in a molecular biology lab. And also at the Weizmann Institute, I started with molecular biology, and I was so interested in that. And I loved the lab I was at the beginning that enabled me, and that was Moshe Oren's lab. Really, like, I feel like it's a founder effect also on biological thinking and experimental strategies, and just by having this confidence that I'll perform my master's with Moshe Oren and Weizmann, we have rotations. So I went wild with my rotations, and that's how I came to Uri Alon's lab, which is a physicist that studies systems biology and trying to find design principles in biology. I said, okay, I'm sure I'll perform my master's on molecular biology, and someday I'll get to bacteria. But let's try to see what is systems biology. So I was always like triggering my curiosity and trying to enrich my background and skills and ways of thinking. At the end, I fell in love with the systems biology, and Moshe was amazing to support this. So my master's was performed in Uri Alon's lab, and also my PhD studies in his lab. Actually, when I transferred from master's to PhD, I asked Uri to switch to microbiology in his lab, but Uri didn't like this idea because his microbiology studies was more like physicist way of thinking of microbiology. And he wanted the biologist to be in his cancer, protein dynamics, and more experimental heavy part of his lab. So Finally, for my postdoc, I was able to pursue this dream to study bacteria and microbes, and developing the thoughts of where and what idea to pursue in my postdoc. I thought that the gut microbiome is just a fascinating field because it's so many microbes living together, these unicellular organisms communicating between each other. The communication is really key in my interest, and also complementing host functions. And that's how I came to Dennis Kasper's lab. I will mention that this talk by Margaret McFall-Nye and on the squid, on the Hawaiian squid and its organ of microbes that help its functions, help the squid to go out at night to look for its prey while hiding from its predators, and the molecular aspects that I learned from a talk by Bonnie Bassler really also shaped my thoughts. So both Bonnie and Margaret also are part of this.
Good, yeah, very, very nice. So one can clearly see and feel literally that you follow your dream, and the dream of the understanding how the microbes really work. That's fantastic. I think that's part of a success. You have to have a dream and you just follow it, and wherever you end up. What is the fundamental insights or the breakthroughs what your work has so far produced?
Yeah, okay. So maybe I'll continue to connect it to my postdoc. So I came to Dennis's lab, and the project that we built together was, I feel, a combination of this systems biology way of thinking with the microbiology and immunology, which was new for me, the immunology especially, and in collaboration with Christoph and Benoit and Diane Matthew that are amazing immunologists. We built this project on microbe-host interactions. And that is a key, I feel. So it's true what you mentioned earlier, that it's foundation of the continuation of the studies in my lab, because the main focus is this interface, the communication between the microbes and the host. Mostly looking at the host immune system, very much looking at the mechanisms of these interactions. We love dynamics. We love to develop tools to enable us to follow this dynamic. One of these tools was developed during my postdoc on fluorescent labeling of anaerobic gut microbes. And in lab, we're also further developing this tool and other tools to enable us to capture microbes in real time while responding to the host and while communicating with the host. And one of our major interests is the functionality of the bacteria. So we see that different bacteria from very further apart phylogeny origins can perform very similar host immunomodulatory functions, and also vice versa. Bacteria that are very similar in the evolutionary aspect or taxonomy can perform even functions on the host, very opposite even functions on the host. And now we're delving into the strain level, identifying whether different strains of the same bacteria are similar or different, and in what aspects are they different, and inside the strain, is there a variability in the population at the strain level? What determines this variability? What are the inputs that cause the trigger to change their phenotypes? Is it reversible? And how does this complement host physiology? I think these are the major studies in lab.
Yeah, thank you so much. I think it's truly, and this is your important contribution, I think, that you are pointing to the complexity of microbial community and the functionality, and it doesn't really matter which species we are talking about. It's the function, and its species are more complex than we think, and strains, and you talked about individuals even within a strain. So I think this is really very important, and this will also be in future work. It will have its impact, that's for sure. Now, Naama, you are a typical example for me where, you are crossing between immunology and microbiology and then system biology per se. And the question is very obvious. How important is interdisciplinary work for you? This is certainly not something what I think a classical trained microbiologist by herself could do, I guess. But my question would be, how important are these different disciplines and integrating the different disciplines in your work? How important is that for your work?
And the nature of our work is very interdisciplinary. I think it enables us to think widely and also deeply. So we think about what's interesting for us, and then we think which tool can enable us to study it. And I think it allows a lot of flexibility in the research. So I like it a lot, but I really endorse also less interdisciplinary scientists that are very focused on specific methods and go deep into their methods. And it's also, I think, very important for science as well. I think my nature is very interdisciplinary in everything in life, I guess. So that's how it's combined also to research. So I like it a lot, and I think it removes barriers. But I also endorse the flip side of less interdisciplinary research.
Yeah, thank you very much. Coming back to your work in the mouse model, we all work on models, and it seems that you are really on an important concept in terms of functionality of the microbiome. The question I'm interested now is how informative is what you find from the mouse gut to the rest of the animal kingdom, including humans, or how are these concepts which are really evolutionary conserved, or should we think of each model system, each model organism has its own messages? I visited your homepage recently, and you have a new press release, and it's called Evolutionary Secrets of the Microbiome. Evolutionary secrets. So now, Naama, how conserved are the mechanisms which you are observing? What do you think, and what do you consider an evolutionary secret?
Yeah. Well, in press release, like in press release, but the evolutionary secrets in that was on this type 1 restriction modification system. It means the idea there was that we found that, we're not the first ones to find that bacteria have inverted repeats that allow flipping of their DNA, and that affects their functionality. So in this system that we studied, it was I feel like it was a core system in this bacteria, in Bacteroides fragilis, that can switch between different transcriptional programs that we saw for sure. And we believe that since we found that it also controls expression of polysaccharides, we believe that also it affects its functionality and the way it communicates with the host, and perhaps also with its neighboring bacteria. So that was this press release, and I would say that it was studied in a mouse model, but with human bacteria.
So here we are. Yeah. So we cross species, and you humanize the mouse with the human microbes, and then they study the function there. That certainly is a good way to get to evolutionary conserved concepts and rules and insights. Yes. As a senior gray-haired person in this field, so if I look back historically to the symbiosis community, that was a very cozy, small number, of investigators around the world, and they studied corals and maybe hydra and some other symbiotic animals, symbiotic partnerships. Now, host-microbe interactions, and we have Gordon conferences, and this is an emerging field. It's really just growing. What is so fascinating there? Why is it so fascinating also for you to be part of that lively community?
Yeah, great question. I think it's, I moved to this field because of like a really personal, professional interest in bacteria and in humans. So that's how I moved to this field. It became, that was 2010. It was already the start of the hype. There are many, many, many more and growing field. And I think it's great to have this growing field because really everyone brings their own aspects and their own angle of looking at things. And I think it's a fascinating field because it's this symbiosis, these inter-kingdom interactions of these ancient bacteria with us humans, and somehow we found a way to depend on each other and help each other live a healthy life. So I find these principles very fascinating. But I'm really happy that the world is studying this field in many different aspects, and I think it's really complementary and synergistic.
Yeah, thank you so much. Now let's move a little bit, if you allow, to your personal professional career. You're a successful investigator. You have your own group now at the Technion in Israel. You are in the science already for quite some time. You made specific decisions why to move from there to there, or joining this lab or not that lab. The question would. And in addition, as I know, you have a functioning family and you have kids. So, Mike, the question, and I think many young listeners which are out there and now listening to this interview here, are interested in learning from you what in your mind is necessary for a successful and a satisfying career in this basic research field.
I think the short answer I would say is in Hebrew, we say to love the way, the path, not to think about the end too much.
Very nice. Very good. Okay. Very nice. Yeah.
That's something very important, especially in science, because so many things, we have so many failures, actually. We have more failures than success. Most of us, maybe I can say so I think if you like the path and the day-to-day, if you wake up happy to your life, then everything else comes along.
That's a very short but a very nice phrasing of I think you can only be successful if you really love and if you're really super interested in what you're doing. And I think it's, that's an important message. I mean, you are based now in Israel, but you moved as a postdoc to Harvard and you returned back. And Israel is not so big, but you changed from Tel Aviv to Haifa. And how important is, and young people always are concerned and interested in the question, how important is mobility? I think it is important. I think in Israel, it's really important in a scientific academic career to move abroad for your postdoc in order to bring back to this small country another field, or usually it's not a field, but another angle in a field. So this is almost mandatory, maybe 98% mandatory in order to have an academic career in Israel. So I think it's really important, the mobility. Inside Israel, it was
kind of depending on the stage and again the interest and the environment. So try to find environments that are very supportive and also insightful and like feeding each other in a supportive manner to pursue our research and our interest in it's really important to have a supportive and happy environment around you. So try to have that throughout.
Thank you so much. That's I can fully understand that. That brings me, of course, to the next question. I mean, if you think, and I fully agree and support what you say, that is mobility is important, that you have to intrinsically be driven what you're doing. Mm-hmm. But you also have family, and this also must be taken into account. But on the other side, you say mobility is important. The question is how you. I have two parts of one question. How do you cope with that? How do you manage to? And obviously, you did manage with that. And what do you expect from an institution? You returned now, or you are now at the Technion. And you have there some time for building your group and making great discoveries and maybe moving on and getting a bigger group. What do you expect from an institution to support this way of a professional career?
Yeah, it's an important point because we in Israel, I know that the scientific community looked at the percentages of women versus men and found that the bottleneck is the postdoc because many women decide not to go for a postdoc because usually we already have a kid or two in Israel before we move to the postdoc. So it's already heavy, husband and kids and accommodating everything and having everyone happy in the career of the spouse. So in Israel, when they realized this bottleneck, they founded fellowship for the spouse. Mm-hmm. So the researcher received the fellowship, but it's actually for the spouse to enable the spouse to have a little bit of oxygen until they found their career path or job for the time of the postdoc abroad. So I think that was really important. Another thing in our case, actually, we just talked about it yesterday at home, was to think about it in early stage. So bachelor's is very early. Then I started my master's and I said, okay, now I'll understand what is research, what's an experiment, how do you get to start and end the project and publish a project. And somewhere in the middle, I understood that I really love it. And that meant, okay, that means I would like to pursue a PhD. And that already means that in 4 or 5 years, I would like us to move abroad. In my case, I was already, I guess, and yeah, at the end of my master's, I think I got married. So it was really an early, and we were already 4 years together or something like that. So it was really in early stages of our partnership that it was clear to me and accepted by Amnon that we will move abroad. It doesn't mean that everything was easy, and Amnon had to take a step or a couple of steps back in his career. And actually, he was in the middle of an internship, and he actually decided to stop the internship in order to have us move because he felt like it's the good timing for me. And of course, that affected and influenced his career. But I think he's very happy with it and was happy to stay. We were 6 years abroad and stayed there. And we all the family feels that we got a lot of enrichment from this. So we had 2 kids before we left and another one over there and another one when we came back. So we came back to Israel pregnant. I was pregnant and a year later.
Yeah, that's so wonderful and so amazing having such a successful, energetic colleague here in chatting with me and knowing there are 4 kids, a happy family. So that's the personal side, but I understood now from you that, of course, the institution you choose to go there also must support that. It's not only on the family side, which is then investing. There must also be investment of the institution, and I understood. I think institutions around the world now realize the problem. So dual careers. But this fellowship program for the fellowship program for the spouse, that's certainly something which we don't have here, and this is something maybe we can learn from your institution here. It's clear message for anybody who is in the administrative level of a university that we have to do something to support exactly what you have, a happy family, a large family, but still being very productive and competitive in your profession. And thank you so much.
Maybe I can add that sure. Also when we looked for a job in Israel and in the world, the institutions were asking, what does your spouse do? How can we help? And so I think the awareness is there. And when we decided to join the Technion, even after I was accepted and started, they continued to ask, what's up with my spouse and did he find a job? And they actively helped looking for a job for my spouse in Israel after we came back. So I think I agree, it's very, very important, the institutional support.
Yeah, thank you so much. I think we can really learn, and I hope that our listeners here outside can also get the message. This is such an important issue these days. We are nearly coming to an end, Naama, of our little chat, but there must be one last question, and this is, of course, because we are not together. You are not here physically, our guest. We cannot go out for dinner tonight together. Which I would love so much, and which we probably most likely will do at some point. We are living in a pandemic, and my question would be, how much does this affect you, and how do you cope with that? All this, Israel was very heavily affected. It's now, what we hear from the news, are doing quite okay or very good. How does the pandemic affect your professional life?
So I'll answer, and then you stop me if it's too long. Of course, the pandemic affected my life, both personally and professionally. The kids are a lot at home, homeschooling, and the lab was locked and then opened partially, and everything is Zoom. Even if we are in lab, each one in its cubicle and office, and we Zoom together. So it definitely affected, but we did all we can to ease the pain of the pandemic. I think Zoom conferences are really good. Of course, it's not the real thing, like the dinners and the mingling and the talking and it's so important to meet physically. It did open up options. Like yesterday, we had a joint lab meeting with a lab at the US. So we wouldn't have thought about it a year ago, although it was possible also then. In the first lockdown, it was like, and we sat at home and tried to understand how do we cope with this, the lab. And we started working on this diagnostic, detection of the SARS-CoV-2 from human samples. But it was really like off the shelf, several products and some tweaking. And it was quite an adventure though, because the lab was really interested, although we were locked at home. So you can also enjoy it a little bit, but we were anxious to do something with the biological pandemic. It's like impossible that we are sitting at home. So after brainstorming and thinking, what can we do? We thought about this method, and the development itself was really quite an adventure. The hospital at Rambam immediately thought it's a good idea or a potential, and we were left to work there with human samples, and we just came to lab and then didn't go back for a week or 10 days or came back only for a few hours. We really like worked 24/7 because we felt like the world needs to know about this method and do something with it. So that's one aspect. Now there is a startup company that took it, moved forward with it to implement it, hopefully to the population. So that's one aspect. The other is this, for sure it slowed down in some aspects. We are missing the human contact, both like having dinner with you tonight and even in lab. We do come to lab most of the week, but still it's not the same as the dinner picture I showed in the seminar in the last slide. So trying to cope, it for sure is not easy.
Yeah, that's true. I fully agree. And but you do amazingly well, and I feel the energy and how you reacted to the COVID by working in the lab, trying to support, and even now giving that to a startup company. That's fantastic.
Thank you.
Very nice. Naama, we are at the end of this wonderful conversation, and thank you so much for sharing your personal views with us. I think our listeners will appreciate and will learn, and this is of great interest. I thank you very much, and I wish you for your further career all the very best. I do hope that we see us here in my institution, that we may start a collaboration, and I do hope for sure that we see us in our joint CFAR family at some point in real meetings again. Thank you so much for your time, and goodbye and very best wishes.
Thank you. Thanks so much for this wonderful time and really great and interesting interview. And I really hope also to come to Kiel, to work together, to start our collaboration, and also to meet in CFAR. I agree. Thank you very much. And lots of success to you too.
Bye-bye. Bye-bye.